The discovery of penicillin stands as a pivotal moment in human history, a triumph of science that has saved countless lives and reshaped the course of medicine. This breakthrough, born from the curious observation of a contaminated petri dish, would go on to revolutionize our understanding of infection and disease. As we embark on this journey, we find ourselves at the threshold of a fascinating tale that will forever change our perspective on the delicate dance between humanity and the microscopic world. In the early 20th century, the world was on the cusp of a profound transformation in the field of medicine. The discovery of X-rays by Wilhelm Conrad Röntgen in 1895 had marked the beginning of a new era in medical diagnostics, and the work of scientists like Louis Pasteur and Robert Koch had laid the foundation for a deeper understanding of microbiology. Yet, despite these advances, the treatment of infectious diseases remained largely ineffective, with doctors often resorting to crude methods such as bloodletting and purging. It was against this backdrop that Alexander Fleming, a Scottish biologist, found himself working at St. Mary's Hospital in London. Fleming, who had served as a surgeon in World War I, had developed a strong interest in the study of bacteria and their role in disease. In 1928, he joined the staff of St. Mary's, where he worked under the guidance of Sir Amroth Wright, a renowned expert in vaccine research. Fleming's laboratory, located on the top floor of the hospital, was a modest affair with limited equipment and a small team of assistants. Despite these constraints, Fleming was a meticulous worker, known for his attention to detail and his ability to think creatively. He spent long hours studying the properties of bacteria, searching for new ways to combat the infections that were then rampant in hospitals. One fateful day in September 1928, Fleming returned to his laboratory after a 2-week vacation, only to find that one of his bacterial cultures had been contaminated with a mold. The culture, which was growing on a petri dish, had been left uncovered, allowing spores from the surrounding environment to settle on its surface. To Fleming's surprise, the mold had formed a visible growth, and the bacteria in the surrounding area had begun to die off. The mold, which would later be identified as Penicillium notatum, had been accidentally introduced into the laboratory through an open window. The spores of the mold, carried on the wind, had landed on the petri dish, where they began to germinate and grow. As Fleming observed the contaminated culture, he noticed something remarkable. The mold seemed to be producing a substance that was lethal to the surrounding bacteria. Over the next few days, Fleming carefully observed the behavior of the mold, noting its effects on different types of bacteria. He realized that the substance produced by the mold, which he later dubbed penicillin, had the potential to revolutionize the treatment of infectious diseases. With this discovery, Fleming had stumbled upon something truly remarkable, a natural antibiotic that could selectively target and kill bacterial cells without harming human tissue. As news of Fleming's discovery began to spread, it caught the attention of other scientists working in the field of microbiology. One such scientist was Howard Florey, an Australian-born pharmacologist who was then working at the University of Oxford. Florey, who had a deep interest in the development of new medicines, saw the potential of penicillin as a therapeutic agent and began to collaborate with Fleming on further research. As Fleming and Florey began to collaborate, they realized that the production of penicillin on a large scale would require significant resources and expertise. Florey, with his background in pharmacology, took the lead in developing methods for mass-producing and purifying the substance. He established a small laboratory at Oxford, where he worked tirelessly to refine the extraction process, often using makeshift equipment and relying on the help of his colleagues. One of these colleagues was Ernst Boris Chain, a German-born biochemist who had fled Nazi Germany in 1933. Chain, an expert in enzyme chemistry, joined Florey's team at Oxford and played a crucial role in developing a method for concentrating and stabilizing penicillin. Together, Florey and Chain worked to optimize the production process, experimenting with different solvents and techniques to isolate the active compound. Meanwhile, Fleming continued to work on the clinical applications of penicillin, conducting experiments to determine its efficacy against various types of bacterial infections. He also began to share his findings with other scientists, presenting his research at conferences and publishing papers in scientific journals. One such paper, published in 1929 in the British Journal of Experimental Pathology, described the antibacterial properties of penicillin and sparked widespread interest among microbiologists and clinicians. As news of penicillin's potential spread, the team at Oxford began to receive inquiries from hospitals and research institutions around the world. Florey and Chain started to supply small quantities of the substance to these organizations, often in the form of crude extracts or imperfectly purified samples. Despite the challenges of working with a relatively unstable compound, the early results were promising, with reports of successful treatments for infections such as septicemia and pneumonia. In 1941, Florey and Chain decided to conduct a series of clinical trials to test the safety and efficacy of penicillin in humans. They chose a group of patients at the Radcliffe Infirmary in Oxford, all of whom were suffering from severe bacterial infections. The results were nothing short of remarkable. Within days of receiving penicillin treatment, many of the patients began to show significant improvements, with some making full recoveries from conditions that had been deemed incurable. The success of these trials caught the attention of the British government, which recognized the potential of penicillin as a game-changer in the treatment of war wounds and infections. In response, the government established a special committee to oversee the development and production of penicillin, with Florey and Chain at its helm. The committee worked to coordinate the efforts of scientists, clinicians, and manufacturers, aiming to produce sufficient quantities of penicillin to meet the demands of the military and civilian healthcare systems. As the British government's committee worked to coordinate the production of penicillin, Florey and Chain faced numerous challenges in scaling up the manufacturing process. One of the major hurdles was the lack of suitable equipment for large-scale fermentation and extraction. The team at Oxford had been using makeshift apparatuses, such as bathtubs and laundry tubs, to cultivate the mold and extract the penicillin. However, these improvised setups were not sufficient for producing the quantities needed to meet the demands of the war effort. To address this issue, Florey and Chain turned to the British company Imperial Chemical Industries, ICI, which had expertise in large-scale chemical production. I agreed to collaborate with the Oxford team and provided access to their facilities and equipment. The company's engineers worked closely with Florey and Chain to design and build specialized fermentation tanks and extraction equipment, enabling the production of penicillin on a much larger scale. Meanwhile, the clinical trials of penicillin continued to yield impressive results. In 1942, a team of doctors at the Radcliffe Infirmary in Oxford, led by Dr. Robert Pulvertaft, conducted a series of experiments using penicillin to treat patients with severe infections. One notable case involved a 15-year-old boy who had developed septicemia after scratching his face on a rosebush. The boy's condition was critical, and conventional treatments had failed to improve his symptoms. However, after receiving penicillin injections, he made a rapid recovery, with his fever subsiding and his wound beginning to heal. The success of these clinical trials helped to convince the medical community of the efficacy of penicillin. As news of its potential spread, hospitals and research institutions from around the world began to request supplies of the substance. The Oxford team, however, was still struggling to meet the demand due to the difficulties in producing sufficient quantities of high-quality penicillin. In an effort to increase production, Florey and Chain established a network of laboratories and manufacturing facilities across the United Kingdom. They also collaborated with other researchers, such as Professor Paul Fieldes, who had expertise in microbiology and biochemistry. Fieldes, who worked at the Wellcome Research Laboratories in London, played a crucial role in developing new methods for assaying and standardizing penicillin, ensuring that the substance was of consistent quality and potency. As the production of penicillin expanded, the Oxford team began to receive visits from prominent scientists and medical professionals. One such visitor was Dr. Charles Fletcher, a renowned physician who had served as a consultant to the British Army during World War I. Fletcher was impressed by the potential of penicillin and recognized its significance in the treatment of war wounds and infections. He became a strong advocate for the substance, helping to promote its use among the medical community and securing funding for further research. As the war effort continued, the demand for penicillin only increased, and the Oxford team worked tirelessly to meet the needs of the military and civilian healthcare systems. In 1943, a team of American scientists, led by Dr. Andrew J. Moyer, arrived in Oxford to learn from Florey and Chain about the production of penicillin. The Americans were impressed by the British efforts and recognized the potential of penicillin to save countless lives on the battlefield. Moyer and his team returned to the United States, where they established a large-scale production facility at the Northern Regional Research Laboratory in Peoria, Illinois. The American effort was led by the War Department's Committee on Medical Research, which worked closely with pharmaceutical companies such as Pfizer, Merck, and Squibb to produce penicillin on a massive scale. The committee also established a network of laboratories and testing facilities to ensure the quality and potency of the penicillin being produced. One of the key figures in this effort was Dr. Selman Waksman, a microbiologist at Rutgers University, who developed a method for mass-producing penicillin using a strain of mold that was more productive than the original Penicillium notatum. As the production of penicillin increased, the substance began to be used more widely in military hospitals and on the battlefield. One notable example of its use was during the Allied invasion of Sicily in July 1943, where penicillin was used to treat wounded soldiers with remarkable success. The drug was also used to treat civilians, particularly those affected by the bombing campaigns in Europe. In London, for example, penicillin was used to treat victims of the V-1 and V-2 rocket attacks, which had caused widespread destruction and injury. In 1944, Florey and Chain traveled to the United States, where they were hailed as heroes and received numerous awards for their discovery. They met with American scientists and politicians, including President Franklin D. Roosevelt, to discuss the production and distribution of penicillin. The visit helped to strengthen the alliance between the two countries and paved the way for further collaboration on medical research. As the war drew to a close, the use of penicillin became more widespread and its impact on public health began to be felt. In 1945, the first commercial production of penicillin began, with pharmaceutical companies such as Pfizer and Squibb marketing the substance under various brand names. The discovery of penicillin also spurred further research into antibiotics, leading to the development of new drugs such as streptomycin and tetracycline. In the aftermath of the war, Florey and Chain continued to work on the development of penicillin, refining their methods and exploring new applications for the substance. They were awarded the Nobel Prize in Physiology or Medicine in 1945, along with Alexander Fleming, for their discovery of penicillin. The award recognized the significance of their contribution to medicine and paved the way for further research into antibiotics. The widespread use of penicillin also led to concerns about its misuse and overuse, particularly in the context of agriculture and animal husbandry. In the late 1940s and early 1950s, scientists such as Fleming and Florey warned about the dangers of antibiotic resistance, which they recognized as a potential consequence of the overuse of penicillin. Their warnings were largely ignored at the time, but they would later prove to be prophetic. In the years following the war, penicillin became a staple of modern medicine, used to treat a wide range of infections and diseases. Its discovery had revolutionized the field of medicine, saving countless lives and transforming the way that doctors treated patients. As the years went by, however, the challenges posed by antibiotic resistance would become increasingly apparent, and scientists would be forced to confront the unintended consequences of their discovery. The discovery of penicillin marked a profound turning point in human history, ushering in a new era of medical advancement and saving countless lives. Thank you for joining me throughout this historical journey. That's all for today's History with Herodotus. Remember to like, share, and subscribe. And until next time, remember that, to understand the present, we must first understand the past.